Hi guys! Today we are going to do plant tissue culture at home. So these are the materials and apparatus needed which you can find in the house. So you need fertilizer to induce the culture growth, agar for the medium, Clorox for disinfection, distilled water, and of course plant to do the tissue culture. So other materials are hand sanitizer, scissors, pressure cooker which will act like an autoclave, plastic wrap, paper towel, tweezer, and also microwavable container. Without further ado, let's do it guys! Oh god, I'm so excited! I hope you guys too! There are three main steps, which are preparation and sterilization of workplace and tools, preparation of the agar, and tissue culture preparation. Let's sterilize the apparatus as well as our workspace. I'm using a pressure cooker to sterilize our apparatus such as container, scissor, and tweezer. And for the workspace, I spray with disinfectant to maintain a sterile environment. So, the second step is agar preparation. Basically, we are adding about 37.5 grams of fertilizer to 750 milliliters of filtered water. And then, we add agar as a solidifying agent. Then, put it in the oven to completely dissolve the agar and to sterilize it. If you have a microwave at home, it is advisable to use that. Now, we have set up our sterile workspace with a mini fan, sample plant, and the sterilized tools that are needed for the experiment. Don't forget to put them on sterilized paper towels. So this is our agar medium, and in this cup is 50% Clorox. Next to it is the filtered water. This setup resembles the laminar airflow. So as you can see, the fan will drive out or prevent any microbes from landing on the workspace. Before we do our experiment, always remember to sanitize your hand using hand sanitizer or any disinfectant and wear your face mask. Afterwards, we spray our medium container. Even it might seem to be perfectly clean, but the outside can be contaminated during the transfer from the oven. Now, we have already cut our sample plant into smaller pieces. So, we dip the plant into 50% Clorox for 10 minutes to remove all the microorganisms that might be deposited on it, such as bacteria and fungi. It is already 10 minutes, so we can take out the plant sample and dip it 3 times into filtered water to remove the excess Clorox before we cut the sample for about 1 cm to obtain the explant. Then, put the rounded part of the explant onto the prepared medium. Now, it is better to just slightly open our container lid to avoid contamination. So last but not least, we need to wrap the container with plastic wrap, also to prevent contamination. Remember, it takes only a single cell of bacteria or spore to contaminate your sample and ruin the whole experiment. You need to keep that in mind. Then place the container under sunlight to promote growth. And that's it! We hope the explant will successfully grow. That's all for now. But please note that the result might be different from experiments done in the lab. However, please always remember to minimize the chance of contamination by practicing precaution steps. Stay curious and happy learning. Goodbye.